Welcome to the video on Chapter 2.8 on cell respiration. So before we start, we need to clarify a couple of terms, okay? So I know in a lot of classes we hear respiration as like respiration rate, and we tend to think that that means breathing, okay? For our purposes, for biology, respiration is not breathing, okay? Respiration is a biochemical process that converts the chemical energy in glucose into ATP. So it's something that happens on a cellular level. We actually don't use the word respiration for breathing. In biology, we use the word ventilation for breathing, and that's the act of inhaling and exhaling. And then when those gases then diffuse out of your lungs and into your bloodstream, we call that gas exchange. Are they linked? You betcha, because we use those gases for cellular respiration, and we have to get rid of them through ventilation. But respiration is not breathing. They're very different. So part of our metabolic processes involve releasing energy in these organic molecules. So I'm thinking organic molecules, okay, like carbohydrates, fatty acids, etc. The energy that's stored in those molecules is in the form of chemical energy, and chemical energy is energy that's stored in the bonds of molecules. A lot of people refer to this as bond energy, and to me that makes way more sense, okay? But in the bonds of molecules, there exists a certain amount of energy, and our job in cellular respiration is to release that energy. So how do we do that? Well, we do that by breaking those bonds, okay? So if we have energy stored in the bonds, then we need to break it to get it out, kind of like breaking a piggy bank to get out the money that you've stored in there. When we do that, okay, that releases energy, but some of that energy is lost as heat, okay? So this isn't an entirely efficient process. Take ATP, for example. You should be familiar with ATP by now, but adenosine triphosphate is our major storage molecule. This is going to be your new best friend in biology because we're going to talk about this all the time, okay? So here's ATP. This right here is a high energy bond. So if we want to get the energy out of ATP, we're going to break this bond. When we do that, we no longer have ATP, we have ADP, adenosine diphosphate, plus that little phosphate group that we broke off. Breaking that bond releases energy. We can use that energy for a whole crap load of things. It's just not entirely efficient. Some of that energy is released as heat, and that's what we call it burning calories. When we break those bonds, okay, it releases heat. When we break bonds to remove or harness some of that energy from a molecule, that process is called oxidation. Okay, so we're kind of like stealing the energy from a molecule, and that process is called oxidation. It can happen at various speeds. Okay, so we're going to talk about two speeds, okay, rapid and slow. So rapid oxidation is exactly what it sounds like. It's releasing all of the energy, that's the oxidative part, really fast, and that's the rapid part. That's something like burning, okay? So when you do that, you're releasing all of that energy really fast, okay? And when we do that, a lot of that energy is lost as heat and sometimes even light, okay? So we are removing that energy in such a quick way that much of the energy is lost as heat and light. That doesn't mean it's a bad thing. Sometimes we like that kind of stuff, okay? But in our cells, from a biochemical standpoint, slow oxidation is much more efficient, okay? And so this is what we're going to use in cell respiration, is slow oxidation. So here we're using enzymes. Remember, enzymes is always the answer, okay? To break bonds and to oxidize molecules one at a time in a very controlled manner, okay? And that's because it's a lot more efficient when we just pick apart those molecules through the use of enzymes, okay, and harness that energy a little bit at a time. Okay, so here is the ultimate goal of cellular respiration. You need to remember this whole thing, but there are some really important points in here. The ultimate goal of cell respiration is to release the chemical energy that's stored in the bonds of glucose in a controlled way, and we're going to try to trap that energy to form ATP molecules. So again, ATP is our major energy molecule. To get the energy out of ATP, we're going to hydrolyze or break this bond, and that's going to release energy for the cell to use. But then we no longer have ATP anymore, we have ADP. So this is like using a credit card, okay? You get all the money out of it, but now you just have an empty credit card. We don't throw it away, we don't get a new credit card, we just recharge it with more money. ATP works the same way, okay? In order to reform this bond here, we have to add energy. This is an anabolic process, and that requires energy. We get that energy by releasing chemical energy in the bonds of things like glucose in a very controlled way so that we can then use that energy in glucose to reform ADP into ATP. So for our purposes, we're going to limit our discussion to the use of glucose as the starting molecule for cell respiration. It's not always the case, okay? We don't always have glucose immediately available. If we don't have glucose, remember glucose is plan A. If we don't have glucose, we can use fatty acids or amino acids, but that's really kind of a last resort. Okay, so let's talk about this word glycolysis. Gly meaning sugar, okay? Lysis meaning to break. So we've seen the Lactic cycle, hydrolysis, okay? That literally means to break. And glycolysis translates into sugar breaking. Here we're going to be breaking down glucose, and it's the first step in cellular respiration. It takes place in the cytoplasm, not the mitochondria. Glycolysis is over here in the cytoplasm or cytosol. So if I have a cell, okay, and here's my nucleus, and then I have some mitochondria. Glycolysis is happening in the cytoplasm, that cytosol jelly water-based stuff. It is an anaerobic process. So what does that mean? Well, that means it takes place without oxygen. Aerobic means with oxygen. I think of aerobics and they need to breathe a lot and they're breathing to get oxygen. But anywho, glycolysis is anaerobic. So we're here in the cytoplasm without oxygen. Okay, I'll work with you on drawing this process in a couple minutes, but for now, let's just outline the steps. So we're going to start with glucose, and glucose has 6 carbons. An enzyme, here's that word we're going to see a lot. An enzyme modifies the glucose to make it unstable. Okay, so glucose on its own is a relatively stable molecule. In order to be able to break it apart, which is the ultimate goal of cell respiration, first we kind of have to destabilize it. Then a series of reactions, which you'll get into if you're on higher level, lucky you, is going to split that glucose into 2 molecules of what we call pyruvate. So if we have 6 carbons and we split it into 2 molecules, that leaves us with 3 carbons in each molecules. The energy from breaking the bonds in that glucose are going to be enough to generate 2 molecules of ATP. Okay, so realistically, the ultimate goal is to break glucose into 2 molecules of pyruvate. Lucky for us, that process also produces 2 ATP molecules. Okay, so let's give this a try, see if we can draw this. So I'm going to start off with glucose, and glucose has 1, 2, 3, 4, 5, 6 carbons. Okay, so here's glucose, and it has 6 carbons. Now, I know you're used to seeing glucose drawn like this, so I could have drawn this in a hexagonal shape, but drawing it in this shape is just going to make our life easier. Okay, so the first step we said is to make this glucose unstable. Now, again, if you get into higher level, we're going to talk about this in more detail, but for now, let's just try to draw this in some kind of unstable looking form. Okay, so here we have unstable glucose. I'm not even going to call it glucose anymore. I'm just going to say it's an unstable 6-carbon thing. All right, and then we are going to end up breaking this into 2 3-carbon molecules of pyruvate. So this gives me 2 pyruvate molecules, and each of them has 3 carbons each. Okay? Now, don't forget, this also gives you enough energy for making 2 ATP molecules. So when we end up breaking this, okay, we're going to be able to generate 2 ATP molecules. Okay, so here's the catch. Okay, to make that glucose unstable, okay, we've got to spend 2 ATP molecules. So it kind of takes energy to get things started. When we break it, that produces 4 ATP molecules. Okay, so we spend 2 to get things started. By the end, we make 4. So that results in a net gain of 2 ATP molecules. Okay, so that's where I'm getting those 2 ATP molecules at the end. Again, we're going from glucose, which is a 6-carbon sugar, to 2 3-carbon compounds, plus a net gain of 2 ATP. Okay, we had to spend 2, so minus 2. We added 4, minus 2 plus 4 gives me a net gain of 2 ATP. Now, cellular respiration is a universal process. All cells do it. All cells must have the ability to harness the energy from organic molecules in a controlled way to make ATP. All cellular respiration pathways in every organism begins with glycolysis. Where it goes after glycolysis is going to depend on a couple of things. It's going to depend on the type of organism, and it's going to depend on the availability of oxygen. Okay? Now, some organisms are able to generate some ATP without oxygen, and they undergo a process called fermentation. You've probably heard of this before. This is the breakdown of organic molecules like glucose for ATP production anaerobically, so without oxygen. This is going to happen in the cytoplasm, not the mitochondria. In order to use the mitochondria, you have to have oxygen. Oxygen's like the gatekeeper of the mitochondria. So since they don't have it, they can't get in there. Anaerobic stuff happens in the cytoplasm only. Now, there are 2 types of fermentation, and the first one we're going to talk about is called alcoholic fermentation. Lucky for us, they're named after what they produce, okay? So the organism we need to remember for alcoholic fermentation is yeast. Yeast is a single-cell organism. It's part of the fungi kingdom. It's a eukaryote, okay? So it has a mitochondria. It has the mechanism for aerobic respiration. Again, it's only going to do anaerobic fermentation if there's no oxygen, okay? So we're going to start with pyruvate, and that pyruvate, that 3-carbon molecule, is going to get broken down into 2 products, carbon dioxide and alcohol, okay? So this is exactly how we produce beer and wine. That's where the alcohol comes from. It's also how we produce bread products. So here's some bread dough. You make bread dough by mixing flour, yeast, and warm water. And you cover it usually with plastic wrap and you allow it to sit. After a few hours, it looks like this. It's all blown up. Why is it all blown up? Well, that's because the carbon dioxide has literally filled up the dough with air pockets. And again, that's why bread has these little air pockets in it. Is there alcohol in bread dough? Yeah, it's the result of fermentation also. Can you get drunk from eating bread? No, because all that alcohol burns off when you bake it. The other type of fermentation is called lactic acid fermentation. And this is what most bacteria and animals do, again, only when they don't have oxygen. Okay? So when they don't have oxygen, they convert that 3-carbon pyruvate into something called lactic acid. Now, this lactic acid can be really useful. This is how we produce things like sour cream, yogurt, cheese. Okay, we use bacteria and we mix it with dairy products and we put it in an anaerobic environment. That produces lactic acid, which makes it taste kind of tangy and tart, and it also keeps it good for much longer than regular milk. Unfortunately, this is also what's responsible for muscle soreness. If you do an exercise where you can get enough oxygen, like walking or light jogging, your cells are going to be fine. They're going to do regular cell respiration aerobically. If you're doing super strenuous exercise, like weightlifting, sprinting, where you literally can't consume enough oxygen to keep up with the aerobic cell respiration process, your cells still need ATP, so they're going to go through lactic acid fermentation. Lactic acid builds up in your muscles after they've undergone this anaerobic lactic acid fermentation, and it causes them to be sore. So that's kind of what you're feeling with those sore muscles. Okay, so let's draw this out. We are, again, starting with glucose, which is our 6-carbon sugar. During glycolysis, okay, that's going to produce 2 molecules of pyruvate. Okay, again, glycolysis happens in the cytoplasm. Okay, if there is no oxygen, we have to stay in the cytoplasm. Okay, and this is going to be fermentation or anaerobic respiration. Okay, depending on the type of organism, whether you are talking about bacteria or animals, or whether you're talking yeast, 2 possible things are going to be produced. Either lactic acid or alcohol and carbon dioxide. Okay, so that's if there is no oxygen, okay, and we're staying in the cytoplasm. If there is oxygen, then we get to go to the mitochondria. Fun times. Okay, and this is where we get aerobic respiration. And every organism produces the byproducts of carbon dioxide and water. Okay, so this is just a little flow chart to kind of help you understand. Everybody goes through glycolysis, where we're taking glucose into pyruvate in the cytoplasm. If there is oxygen, we get to go to the mitochondria. We're going to undergo cellular respiration and produce carbon dioxide and water. If there's no oxygen, we have to stay in the cytoplasm and undergo fermentation. If you're bacteria or an animal, you're going to produce lactic acid. If you're yeast, you're doing alcohol and carbon dioxide production. Okay, so since aerobic cell respiration takes place in the mitochondria, let's take a moment to kind of review how to draw a mitochondria or the parts of a mitochondria. So if you'll remember, mitochondria originated through the endosymbiotic theory, where we had some kind of heterotrophic bacteria being engulfed. This bacterium had its own membrane. It got engulfed via endocytosis and surrounded by a vesicle, which ended up evolving into the outer membrane. So mitochondria have a double membrane. They have an inner membrane from the original bacteria and an outer membrane from that original vesicle. Okay, so we should be drawing our mitochondria with a double membrane. Let's draw that out now. So here's my outer membrane, and then my inner membrane is kind of squishy. It's in these finger-like projections. Okay, so we have the outer membrane. We have the inner membrane. Okay, we have the folds in the membrane, the folds in the inner membrane called cristae. And they're like these things, these folds. Inside of the inner membrane, we have the area known as the matrix. And in between the inner and outer membrane, we have what's called the intermembrane space. And that's the space between the inside and outside membrane. The reason why you're asked to know this is because different parts of cellular respiration are going to occur in different parts of the mitochondria. So basically, cellular respiration goes something like this. We're going to take glucose, C6H12O6, and in the presence of oxygen, so here we're obviously talking about aerobic cell respiration, that's going to get broken down into carbon dioxide, water, and energy. Okay, now that energy, you can make a note of this if you want, I think it's useful, is going to be used for two things. Okay, it's going to be used to generate ATP, but it also accidentally, as we talked about, releases heat energy. This isn't a balanced equation. Okay, these are just listing the reactants and products. Okay, so here are my basic steps. So first, pyruvate, which we got from glycolysis, and oxygen are going to enter the mitochondria. Again, nothing can happen in the mitochondria without oxygen. The pyruvate, that 3-carbon molecule, those are supposed to be 3 carbons. Wow, that's bad. 1, 2, 3. That 3-carbon molecule is going to get completely broken down into reactions called the Krebs Cycle on the electron transport chain. So we're going to go from a 3-carbon molecule, okay, to completely taking it apart. So we're starting with glucose, 1, 2, 3, 4, 5, 6 carbons, and we're breaking it down completely, okay? So by the end of this, we've broken all of those bonds, and as we talked about earlier, breaking these bonds releases energy. Water, carbon dioxide, and ATP are generated along with some of this accidental heat, okay? If we do this aerobically, and again, this is all aerobic, that generates 36 ATP. Anaerobic respiration, like what we talked about earlier in that fermentation process, produces a total of 2 ATPs. Both of these include glycolysis, okay? Glycolysis. So as you can see, this is way more efficient than this. Okay, so our first choice is always to do it aerobically because we can produce more ATP. Anaerobic is only plan B if there's no oxygen available. Okay, so this is not a very long video. You're not spending tons of time on this. So do yourself a favor. Go ahead and pause. Take some time to answer these questions and then start replaying the video again to check your answers. Okay, in the first question you were asked about the germinating pea seeds, and so that's these two. What type of respiration is occurring? Okay, and the answer there would be aerobic. Okay, and I know it's aerobic because they are consuming oxygen. It says right here, this is oxygen consumed, and they are consuming lots of oxygen. You're only consuming oxygen when you're doing aerobic respiration. In the second one, it says, why is the oxygen consumption for non-germinating seeds very low? So here are my non-germinators. Okay, well, the answer to that is because there is no respiration, or I should say no aerobic respiration happening. Okay, so if there's no aerobic respiration, they're not going to be consuming very much oxygen. And then in this last one, we were asked, why would the germinating seeds show a greater oxygen consumption at 22 than 12? So here's 22 versus 12. If something is consuming more oxygen, okay, that must be because it's undergoing a faster rate of cellular, or I should be specific, of saying aerobic cellular respiration. And I'm going to abbreviate there because I'm really tired of writing on this stupid screen, sorry. Okay, so it's going at a faster rate of aerobic respiration, which is why it's consuming more oxygen. And that'll do it for chapter 2.8 on cell respiration, unless you're in higher level, and then we have to do chapter 8.2 on cellular respiration. So enjoy this easy stuff while it's lasting.